good day to my respected lecturer and my fellow friends. I'm Nandini Gopal. So today me and my groupmates are going to present about the application of gas chromatography. First of all, I would like to introduce my groupmates. My groupmates are Tivia Subramaniam, Fadila binti Muhammad Fauzi and also Muhammad Nasrun. Moving to our next slide, which is chromatography. Basically, chromatography is a laboratory apparatus which is involved a sample being dissolved in a mobile phase, which may be a gas, liquid, solid, or what we call a supercritical fluid, and which carries it through a system called a column, a capillary tube, a plate, or a sheet. Basically, there are four types of chromatography. The first one is liquid chromatography, and the second one is gas chromatography. The third one is thin layer chromatography, and the last one, the fourth one is paper chromatography. Here in this slide, I have highlighted the word gas chromatography. Why? Because as I told before, today our main topic is about gas chromatography. Let's take a look on the part which is what is gas chromatography. Actually, gas chromatography is used to determine the chemical compounds, the chemical compositions of unknown substances, such as the different compounds in gasoline shown by each separate peak in the graph. And it also used to separate vaporized sample with a carrier gas in mobile phase and a column composed of a liquid or of a solid beads in stationary phase. And gas chromatography also used in analytical chemistry for separating and analyzing compounds that can be vaporized without decomposition. And it also typical uses of gas chromatography include testing the purity of a particular substances or separating the different components of a mixture. Now we are going to take a look on the type of sample. Basically, there are two types of gas chromatography. The first one is gas solid chromatography. The short form is GSC, and the second one is liquid gas chromatography, and we call it as GLC. Now, the gas solid chromatography is applied to separation of mixtures of hydrogen isotopes, isomers, proteins, or virus, and it also an important analytical tool for the separation and identification of various mixture from hydrogen isotopes and isomers of high boiling point of substances. The gas liquid chromatography, which is GLC, is used in which the substances to be separated are moved by an inner gas along with a tube filled with a finely divided inner solid coated with a non-volatile oil, and each and every components migrate at a rate determined by its solubility in oil and its vapor pressure. This is the last slide, which is definition of technique used in analysis of sample. The technique that used to analyze the sample is analytical technique. The analytical technique is used to separate the chemical compounds of a sample mixture and then detect them to determine their presence or their absence or how much is present. And these chemical compounds are usually organic molecules or gases. That's all from my slide. So the next slide and the next points will be elaborated and explained by my groupmates. Thank you.
Good evening. My name is Tivia. I'm going to present about preparation of sample and methods of analysis in gas chromatography. Let's talk about the preparation of sample. This is a process of transforming a sample into form that is suitable for analysis. In this process, we need to extract the sample and then need to do analysis. There are three methods in this process. First is purge and trap, solid phase microextraction, and thermal desorption. Purge and trap is to remove volatile analytes from liquid or solids, concentrating the analytes and introducing them into a gas chromatograph. It also removes 100% of analytes from the sample. Quantitative removal of polar analytes from matrices can be difficult. Okay, this is the image of the perch and trap. The next is solid phase micro extraction. This method is to extract compounds from liquid, air or sludge without using any solvent. This also permits a selective extraction of the target organic compound not to adsorb by the nanoparticle directly into the reaction vessel, which reduce effect on the interaction between the nanoparticles and probe compounds. The key compound is the fused silica coated with a stationary phase similar to those used in gas chromatography. Okay, this is the process of this method. The final one is the thermal desorption. In this method is to release volatile compounds from solid samples. The sample is placed in a steel or glass tube and held in a place with glass wool. The sample is purged with carrier gas to remove oxygen. Desorption tube is connected to the chromatography column and they heated to release the volatile substances that are collected by cold trapping at the beginning of the column. Okay, the column is heated rapidly to initiate chromatography, that is the picture of the thermal desorption. Let's see the method of analysis in GC. GC means known as gas chromatography. Gas chromatography analysis is known volume of gases or liquid analyte is injected into the heat of the column, usually using a micro syringe. This motion is inhibited by the adsorption of the analyte molecules either onto the column walls or onto packing materials in the column as the carrier gas sweeps the analyte molecule through the column. The rate at which the molecules progress also the column depends on the strength of adsorption, depends on the type of molecule on the stationary phase materials. Since each type of the molecule has the different rate of progression, the various components of the analyte mixture are separated as they progress along the column and reach the end of the column at different times, which is retention time. A detector is used to monitor the outlet streams from the column. Thus, the time at which component reaches the outlet and the amount of that component can be determined. Substances are identified by the order in which they are elute from the column and by the retention time of the analyte in the column. That's all. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Fadila binti Muhammad Fauzi and I will present about suitable parameters used in GC. The first parameters used in GC is retention time. Retention time is the difference in time between the point of injection and appearance of peak maxima. Retention time measured in minutes or seconds. So basically, retention time is the time required for 50% of a component to be elute from a column. Okay, the next one is retention volume. Okay, retention volume is the volume of carrier gas which is required to elute 50% of a component from the column. So retention volume is the volume of mobile phase that is required to elute a substance from a column. So the formula to get the retention volume is retention time times flow rate. The third parameters is separation factor. Separation factors is ratio of partition coefficient of the two components to be separated. So what it means by that is if there is more difference in partition coefficient between two compounds, the peaks will be far apart, and if there is less difference in partition coefficient of the two compounds, so the peaks will be more closer. The next parameters is resolution. The resolution is the true separation of two consecutive peaks on a chromatogram is measured by resolution. It is the measure of both column and solvent efficiencies. So what it means by resolution is it is a quantitative measure of how well two illusion peaks can be differentiated in chromatographic separation. To get resolution, the formula is the difference between two rotation times divided by the combined width of the two illusion peaks. Okay, the last parameters is theoretical plate. Theoretical plate is an imaginary unit of the column where equilibrium has been established between SP and MP. It can also be called as a functional unit of the column. Okay, next we will move to the possible results. Okay, once the experiment is done, the result it shows as peaks on the resulting chromatogram. So from the peaks, we can know all the informations that we needed. For example, what type of compound present in our sample. The compound is more affinity to mobile phase or stationary phase, and the concentration of the compound in our sample, and so on. Okay, the next one is x-axis. Okay, x-axis is the amount of time taken for the analytes to pass through the column and reach the mass spectrometer detector. So basically what is shown in the x-axis is the retention time, which is the time taken for the compound to reach at the detector. So if the compound is more affinity to mobile phase, the retention time will be less. But if the compound is more affinity to stationary phase, so the retention time will be higher, will be more. Okay, the last one is y-axis, or the area of the peak. Is a reflection of the amount of a specific analyte that's present. So basically, at y-axis, it shows the concentration of the analytes that present in our sample. So the higher the concentration of an analyte present in our sample, the higher the peaks will be, and vice versa. Okay, here I have attached an example of result. So as we can see from the graph, it have 3 peaks, which means that the sample have 3 different compounds in it. And the first peaks shows that the compound is more affinity to mobile phase, while the last peak shows that the compound is more affinity to stationary phase. Okay, next we can see that the first peak has the highest peak, which means that the first compound has the highest concentration in the sample, and the least one is the second peak. Okay, so all the information we can get from this graph from the result. Okay, that's all from me. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Asrul bin Amashkali, and I will present about how gas chromatography can can improve the quality of health or environment. Okay, firstly, I will give an example. So, for example, is a determination of pesticides residue in aquaculture products. Okay, first I will talk about agriculture. So, agriculture chemicals such as fertilizers and pesticides have made an important contribution to agriculture. Okay, pesticides protects crops from pests and disease. They have brought about a large increase, and since among all chemicals that can kill pests are also potentially damaging to human health, so the pesticide is important. So we diagnose the compounds in the pesticide by using gas chromatography. Okay, first, legislation requires that pesticide use is appropriately controlled and maximum residue levels (MRL) not be exceeded. So the compounds must be identified and will not exceed that the need of pesticide that can give harmful to the environment or health. Okay, next, the detection method is the process of identifying the pesticides and determining the concentration with the aid of analytical instrument. So we use the gas chromatograph, the instrument that we use to identify the compound in the pesticide. So after that, we can use the gas chromatograph to obtain the information about the original sample by running a standard solutions containing the pesticide at a known concentration and compare it with the sample. So we take the sample of pesticide and we do the gas chromatograph and we compare with the sample to know the concentration whether it's acidic or not, that give the in the legislation requires. That's all from me. Thank you.